Our immunity is what protects us from disease. The word immune actually means to be free from burden, in this case to be free from the burden of disease. A pathogen can cause disease if it can enter our body, we talked about the portals of entry, attach to host cells, we talked about adhesins and special structures that allow organisms to do that, and evade the body's defenses. Those would be those virulence factors, exoenzymes, toxins, and antiphagocytic factors. There are a great many diseases that we are not susceptible to because we have species resistance. There's only a handful of diseases we can acquire from animals, just a few zoonoses. Pretty much everything else we're protected from by being Homo sapiens. Our body defenses can be broken down into what we call the 3 lines of defense. The first line is barriers. Now, this is part of our innate immune system. Everyone is born with the same first line of defense. The second line of defense are some nonspecific internal processes. These are also innate. We are all born with these same nonspecific internal processes. The third line of defense is our specific immunity. This is acquired. This is our adaptive immunity. This can be a little different from person to person because it will depend upon what we have been exposed to in life. Our first line of defense are our body surfaces. This will be things like our intact skin and mucous membranes. The mucus on mucous membranes acts to trap bacteria, and a great many of our mucous membranes have ciliated cells. As the cilia move, it will move the mucus toward a natural opening. Sweat, saliva, urine, and tears all come from natural body openings. These are ways that bacteria can get in. But because these fluids are flowing out, they have a flushing effect on these tubes. Underneath our skin and mucous membranes are a host of phagocytic cells. These are called the dendritic cells. They're there should anything just wiggle through a cell somewhere. Defensins and dermacidins are found in some of our body fluids. Defensins can be found in mucus, saliva, and some of the secretions on our skin. Dermacidins are found in sweat. Lysozyme is found in tears and saliva. This is particularly good against defending against gram-positive bacteria. Your skin oil is acidic. Not a lot of bacteria like living in that environment. And then our body surfaces all have normal flora. These are organisms that are growing there and provide microbial antagonism. If they're taking up all the space, then there's no room for anything else to move in. Should our first line of defense be breached, should there be a break in our skin, then our second line of defense can come into play. Our second line of defense are a group of cells, antimicrobial proteins, and processes that help prevent further invasion. These things will be our defensive blood cells, the chemicals interferon and complement, the process of inflammation, and the process of fever. Part of our second line of defense depends upon the body's ability to recognize invaders. Now, this is not a specific recognition. This is more an invader as you're not supposed to be here, you're not normal kind of thing. To do this, we have toll-like receptors and NOD proteins. These are both involved in triggering second and third line defenses. The toll-like receptors are a group of integral proteins of the cytoplasmic membranes of our phagocytic cells. These act like an early warning system. If these are activated, they trigger body responses to molecules that are shared by various bacterial and viral pathogens, things like flagellin on a bacterial flagellum, or peptidoglycan in a bacterial cell wall, or viral DNA or RNA. These components on the foreign cell are known as pathogen-associated molecular patterns, or PAMPs. When a PAMP binds to a toll-like receptor, it initiates several defenses. The infected cell can undergo apoptosis, we can trigger inflammation, and we can trigger stimulation of the specific immune system. The NOD proteins are like the toll-like receptors, but they're located inside the cell. They bind internally and they will trigger cell death by apoptosis. They also trigger other second-line immune responses. It's believed that mutations in the NOD genes may be associated with inflammatory bowel diseases like Crohn's disease. Crohn's disease was always believed to be an autoimmune disease, but now it may be more a deficiency of the immune system than an autoimmune disorder. Your defensive blood cells are your white blood cells. Basophils are present in the blood in very small numbers. They're the lowest number of blood cells you have. Their granules contain histamine, and histamine is a potent activator of inflammation. Eosinophils have granules that contain antihistamine. The inflammatory process is sort of a positive feedback mechanism, so we have to have an off switch for it, and the eosinophils can provide that off switch. The eosinophils also contain digestive enzymes that are effective against parasitic worms. The lymphocytes are the cells that are going to be involved in specific immunity for the most part. There is a small population of lymphocytes that do not get educated. They're part of our non-specific immunity. These are called natural killer cells. The neutrophils are phagocytic cells. These are the first cells to show up whenever there's an invasion and they begin the eating process, the destruction process. The monocytes are our second-line phagocytes. Now they are larger, they can ingest larger things. They come after the neutrophils. The monocytes also have the ability to leave the blood through diapedesis, and they become macrophages. Now we have two kinds of macrophages in the body. Wandering macrophages are the monocytes that leave the blood, meander through the tissues, return back to the blood. They're looking for things that they need to eat. Some tissues have fixed macrophages. That is, their macrophages are part of the internal tissue. In your lungs, you have fixed macrophages. In your liver, you have fixed macrophages. These cells are always there and they help that tissue protect against invasion. Phagocytosis is a process that helps eliminate bacteria and other foreign cells from the body. It relies on chemotaxis, that is, chemical signals that tell the phagocytes where to go. Positive chemotaxis causes things to move toward them. Negative, they move away. Chemotactic factors are released during a number of processes that attract phagocytic leukocytes. Defensins can act as chemotactic factors, so can certain chemokines, chemicals that are released at the site of inflammation, and PAMPs act as potent chemotactic factors. The process of phagocytosis begins with adherence. The phagocyte has to attach to an organism. This is why a capsule is an antiphagocytic factor. It does not allow adherence to occur. Then the organism is taken in as a phagosome. The phagosome will merge with the lysosome so that the ingested material will be destroyed. Here again, some of those antiphagocytic factors prevent this step from occurring. Anything that's not digested will be eliminated by exocytosis from the cell. Basically, the cell is using this bacterial cell as food. So in the process of phagocytosis, we have adherence, ingestion, digestion, and elimination of anything that wasn't digested. Those natural killer cells are lymphocytes that are not differentiated. That is, they are not taught to look for a particular antigen. They can, however, identify not self cells. They know self proteins and they just go through and say it's self or it's not self. They don't worry about what kind of not self it is. Cancer cells and virally infected cells will put altered proteins on their cell surface. This will allow natural killer cells to identify these as abnormal, not self cells. Natural killer cells will secrete toxins that will kill the cell. Now, natural killer cells, even though they are lymphocytes, are still part of our secondary line of defense. They're not specific for a particular protein or a particular cell type. They only go after something that's not normal. Interferons are protein molecules that help inhibit viral spread. Again, these are nonspecific. They're effective against any virus. There are three kinds of interferon. Alpha and beta interferons are secreted by virally infected cells. Unfortunately, these interferons cannot protect the cells that secrete them. The infected cells secrete interferon, which diffuses to nearby cells. This interferon stimulates the uninfected cell to produce an antiviral protein, an interfering protein, that will inhibit viral replication. Gamma interferon, the third type of interferon, is secreted by T lymphocytes and natural killer cells, and it plays more of a role in regulating immune activity. So here is the infected cell. The virus comes in, takes over the cell. We're going to make a lot of new virus particles. But part of what happens is the interferon gene is activated, and this cell produces interferon. Interferon will be released and will be able to diffuse to a nearby cell. Now, as this cell makes its viral particles, we get all these viruses that are released, and they're going to go to nearby cells and try to invade. When they go in, they find that their replication process is blocked by this antiviral protein that was made as a result of the interferon activating genes for antiviral protein in the uninfected cell. So this is how we prevent viral spread. If the virus can't replicate inside this cell, then we stop the cycle of making more virus particles. Complement is a set of serum proteins that circulate the blood in an inactive form. They have a cascade type of activation. You saw this if you've seen blood clotting factors, how they work. There are multiple pathways to initiate the complement cascade. The classical pathway is triggered by an antigen-antibody complex. This means that the third line of defense has to be in play at this point. An alternate pathway can be triggered by pathogens or pathogenic products, endotoxins, cell wall components. These things can activate the complement pathway. And then the lectin pathway is activated by microbial polysaccharides. They can bind to complement and actually activate the complement sequence. As complement is being developed, as we're going through this cascade of events, we have certain components of complement that can act as opsonins. They can code up bacterial cells, making them easy for phagocytic cells to find and engulf. They can enhance inflammation. But when you get the full complement cascade activated, what they really are out to do is to lyse the cell that started the whole event. The full complement cascade is called a membrane attack complex, and it literally sits down on a cell membrane and pokes a hole in it. So 3 different pathways to basically get to the same thing, this membrane attack complex and cell lysis. Along the way, we have some components that can tenderize the bacteria to make phagocytosis easier. We have a couple of components that will stimulate inflammation and enhance inflammation. But when we get the full cascade, we poke a hole in the offending cell. Inflammation is a nonspecific response to tissue damage. Any kind of tissue damage, heat, chemical, abrasions, cuts, pathogens. Inflammation and infection are 2 different things. Ideally, inflammation is an acute response. It's very quick, it's short-lived, and it's beneficial. However, if inflammation goes on too long and we have chronic inflammation, this usually ends up being harmful. Inflammation is a positive feedback mechanism, so we've got to be able to turn this off. The cardinal signs and symptoms of inflammation are redness or rubor, localized heat, calor, edema, swelling, and pain or dolor. These are the 4 cardinal signs. Many people add a 5th sign here, which is loss of function. Usually the pain and the swelling makes it hard for you to move the inflamed part. We think that loss of function contributes to the inflammatory process by making us rest. As we start inflammation, we're going to dilate and increase the permeability of capillaries in the damaged area. Mast cells from the damaged tissue will release histamine. Damaged tissue cells can also release other inflammatory proteins like bradykinin, prostaglandins, and leukotrienes. Complement may also be activated, and it may be involved in some of this process. The idea is as we open up the blood vessels, we're going to deliver blood cells, which are phagocytes, nutrients, we're going to flush the tissue with fluid, and we're going to bring fibrin into the area to wall off the area because part of inflammation is to contain the damage. The heat, The swelling and the redness, which are signs of inflammation, are all caused by this increased blood supply to the area. Blood is warm, blood is red, and the extra permeability of the capillaries allows more fluid to leave the blood and enter the tissues. The swelling will usually trigger pain receptors, and because of the swelling, you may also have that loss of function. Histamine is the major contributor to this first part of the inflammatory process. An exudate is a result of edema. This is the extra fluid in the tissue. The purpose of this fluid is to dilute any toxins that may be in the tissue. The exudate may drain from an open wound. We classify it as serous if the fluid is clear, serosanguinous if the fluid contains blood, and purulent if it contains pus, and pus is dead tissue cells and macrophages. Inflammation begins with the migration of the phagocytic cells. Chemical signals will cause the release of leukocytes from the bone marrow. This provides us with leukocytosis, or an increased white blood cell count. Then we have the development of cell adhesion molecules, or CAMs. These are generated at the site of inflammation. As the white blood cells flow by, we want to make sure that they stay at this location. So the CAMs act as attachment proteins. As the white blood cells are flowing by, they attach to the CAMs. This is what we call margination. We have the white blood cells lining up along the capillaries. Once the white blood cells are on the walls of the capillaries, they will use diapedesis to enter the tissue. Cytokines from the damaged tissue provide positive chemotaxis so that the white blood cells migrate exactly to the point of injury. The purpose of inflammation is to allow for tissue repair. The macrophages go in and clean up the debris. Damaged cells take care of any bacteria. Basically, they set the stage for healing. The fibrin that was used to wall off the area becomes replaced with collagen. New cells begin to replace the old damaged cells if the tissue is regenerative. If not, the collagen becomes scar tissue. So here we see an injury. And in this case, we have bacteria that are coming in this injury. The inflammatory chemicals start acting as chemotactic agents. We're going to trigger leukocytosis so that extra white blood cells enter the bloodstream. Then we're going to develop those cell adhesion molecules so that the white blood cells stick. This is the margination. Once they're there, they're going to move through the blood vessel and get into the tissue. This is diapedesis. Then we're going to have the chemical signals that will call them exactly to the site of injury. Fever is another of the body's nonspecific defense mechanisms. Fever is a body temperature above 37 degrees centigrade. The hypothalamus controls your thermostat. Now, the hypothalamus can be reset by pyrogens. Pyrogen literally means fire producer or heat producer. Pyrogens are chemicals that are produced by damaged cells or by bacterial cells. They go to the hypothalamus and they reset the temperature controls in the hypothalamus. When your body thinks it's too cold, because now the hypothalamus thinks your body temperature should be 102 instead of 98.6, the body goes through all of the normal heat generating mechanisms. The blood is pulled deep into the organs, so people look pale. They begin to shiver because they're trying to generate heat, so they have the chills. Once the pyrogens disappear, then the heat-reducing mechanisms come into play. Sometimes breaking a fever is called the crisis of the disease. The pyrogens have been eliminated. The hypothalamus resets to 98.6 degrees Fahrenheit, and we begin all of the heat-reducing mechanisms. We sweat. We get red because we bring all the hot blood to the surface. The purpose of fever is to reduce circulating zinc and iron. These can be growth-limiting factors for many pathogens. It won't prevent them from growing totally, it won't kill them at all, but it will reduce their ability to reproduce as quickly. It will lengthen their generation time. By reducing the number of bacteria in the body, it gives the immune system a little longer to get a handle on things. Also, fever enhances metabolism and the effects of the other nonspecific processes. And it makes you feel bad, so you're more likely to rest, which is something you need to do if you're recovering from an infection. Low temperatures are okay, and most physicians like to let low temperatures kind of play out because of the protective mechanisms, but high temperatures can cause organ damage and death.